Branching morphogenesis is the remodeling of the epithelial or endothelial sheaths into multicellular tubular networks. This process occurs in many animal organs, such as the lungs, kidneys, and various glands, including the salivary glands. The salivary gland secretes fluids including water, electrolytes, mucus, and enzymes. These fluids are involved in binding and lubricating food into a bolus and assisting in the overall digestion process.
Along with multiple other functions, the salivary glands initiate starch digestion by secreting an alpha amylase, which begins to digest dietary starch into maltose. In mice, this is extremely important as starch is a major component of their diet.
Most animals have three major parts of salivary glands that differ in fluid secretion. In mice, those three glands are the exorbital lacrimal gland, the submandibular salivary gland, and the parotid gland.
The mouse embryo takes about 21 days to fully develop. The salivary glands begin developing in the prenatal embryo, and they take about 5 days to complete the stages of branching morphogenesis.
The process of branching morphogenesis in the development of the salivary glands begins at about embryonic day 11. At this stage, the developing embryo begins to show signs of advanced cranial facial development, with well-defined palate and peripheral margins of the eye.
The process begins with the pre-bud stage. This stage is characterized by a thickening of epithelial tissue through cell proliferation and growth at the site of origin within the oral cavity. As the epithelial tissue continues to proliferate, it invaginates and begins to grow down into the underlying mesenchymal tissue. This forms the initial bud, a solid stalk of epithelial cells forming the primary duct connected to the oral cavity and terminating in a solid bulbous tip. At this early stage in development, the interaction with the mesenchymal tissue is particularly important as it provides structural support and vital molecular signaling. Progress into the pseudoglandular stage begins with cleft formation on the tip of the initial bud, dividing the main bud into 3 to 5 neighboring buds. Epithelial cell proliferation continues, extending the buds out into branches of their own. The canalicular stage is characterized by a highly branched structural mass of epithelial cells. At this point, there is controlled cellular apoptosis beginning in the center of the buds, resulting in hollowed out lumen and channels within the branches.
Terminal budding is a highly branched stage with fully formed lumens and ducts. At this point, there is a large amount of cellular differentiation depending on the function of that salivary gland. Amongst other things, this differentiation process determines which part of the gland secretes a particular fluid. Also, this differentiation is mostly dependent upon the interactions between epithelial cells, while the extracellular matrix and mesenchymal cells have little influence. The terminal bud stage usually occurs around embryonic day 16. At this point, there is almost complete craniofacial development, and the salivary gland has completed its embryonic development. There will also be additional postnatal development and maturation in the gland.
Salivary gland formation and branching morphogenesis in the embryo is a highly complex process of cell proliferation, growth, and apoptosis. It is dependent on various and varying signaling processes between the epithelial cells, a dynamic underlying mesenchymal cell population, and the extracellular matrix throughout the development of the gland.